Hello viewers, welcome to my channel. In this video, we will discuss some important questions on DNA replication in prokaryotes and eukaryotes. In my previous video, I have already discussed some important questions on DNA replication. The link is shared in the description box. You can check it. For CSR NEET, ICMR NEET, GATE, NEET exams, the questions regarding this topic is very important. Some brief notes regarding the questions and their related options are also provided here, which will be beneficial for you. So stay tuned in this video. The first question. In prokaryotes, leading strands synthesized by option 1, DNA polymerase 3, option 2, DNA pol 1, option 3, polymerase delta and epsilon, option 4, polymerase alpha. And the correct answer is option 1, that is DNA pol 3. In prokaryotic cells, DNA pol 3 is the major replicative polymerase that functions in the synthesis of both leading strand of DNA and of OKAZAKI fragments by the extension of the RNA primer. Whereas in eukaryotes, the leading strand synthesized by polymerase delta. Next question. The length of OKAZAKI fragments generated during lagging strand synthesis in eukaryotes is option 1, up to 200 bases long, option 2, up to 2000 bases long, option 3, up to 100 bases long, option 4, up to 1000 bases long. And the answer is option 1, that is 200 bases long. In eukaryotes, OKAZAKI fragments generated during lagging strand synthesis is shorter than those in E. coli. It is up to 200 bases in case of eukaryotes, whereas in prokaryotes, that is in E. coli, it is up to 2000 bases long. Next question. In eukaryotes, lagging strands synthesized by option 1, DNA pol 3, option 2, DNA pol 1, option 3, polymerase delta and polymerase epsilon, option 4, polymerase delta. And the correct answer is option 3, that is polymerase delta and polymerase epsilon. Polymerase delta is the major replicative polymerase in leading strand synthesis in case of eukaryotes, whereas polymerase delta and polymerase epsilon are the major polymerase in lagging strand synthesis. This is similar to DNA polymerase 1 and 3 in the lagging strand synthesis in prokaryotes. Next question. In prokaryotes, lagging strand synthesis initiated by option 1, DNA pol 3, option 2, DNA pol 1, option 3, primase, option 4 is polymerase alpha. And the correct answer is option 3, that is primase. Primase is the enzyme that synthesizes RNA primers, the oligonucleotides that are complementary bound to a nucleic acid polymer. This primase is required because DNA polymerase cannot initiate polymer synthesis on single-stranded DNA templates, and they can only elongate from the 3' hydroxyl of a primer. Next question. In eukaryotes, lagging strand synthesis initiated by option 1, DNA pol 3, option 2, DNA pol 1, option 3, primase, option 4 is polymerase alpha primase complex. And the correct answer is option 4, polymerase alpha primase complex. The DNA polymerase alpha primase complex synthesizes the first primer. It initiates DNA replication on the leading strand and OKAZAKI fragments on the lagging strand in case of eukaryotes. In addition to the polymerase alpha primase complex, the two DNA polymerase, that is polymerase delta and polymerase epsilon, are required for DNA replication. Polymerase alpha primase complex initiates DNA synthesis in both leading strand and lagging strands by synthesizing a RNA-DNA hybrid primer. The replicative DNA polymerase, that is polymerase delta and polymerase epsilon, that extend the DNA synthesis to form the primer. Note that the polymerase delta is required for the extension of DNA synthesis in case of leading strand synthesis of eukaryotes, whereas polymerase delta and epsilon both are required for the extension of the lagging strands in case of eukaryotes. Next questions. In eukaryotes, which enzyme help in joining after RNA primer removal? Option 1 is DNA pol 3 and DNA ligase. Option 2 DNA pol 1 and DNA ligase. Option 3 polymerase delta and DNA ligase. Option 4 polymerase alpha and DNA ligase. And the correct answer is option 3, that is polymerase delta and DNA ligase. In eukaryotes, the RNA primers are removed by the combined action of RNase H that degrades the RNA strand in the RNA DNA hybrids and a 5' to 3' exonucleases. After the primer removal, the gaps are created, and these resulting gaps then filled by the polymerase delta, and the DNA fragments are joined by DNA ligase, yielding an intact lagging strand. Next question. In prokaryotes, which enzymes help in gap joining after the RNA primer removal? Option 1 is DNA pol 3 and DNA ligase. Option 2 DNA pol 1 DNA ligase. Option 3 polymerase delta and DNA ligase. Option 4 is polymerase alpha and DNA ligase. And the correct answer is DNA pol 1 and DNA ligase, that is option 2. In prokaryotes, RNA primers are removed by DNA pol 1 by its 5' to 3' exonuclease activity. Whereas in eukaryotes, the RNA primers are removed by RNase H. Okay, RNase H plus 5' to 3' exonuclease. Okay, so in case of prokaryotes, the RNA primers are removed by DNA pol 1 by its 5' to 3' exonuclease activity. Whereas in case of eukaryotes, the RNA primers are removed by RNase H plus 5' to 3' exonucleases. The enzymes are different from between for eukaryotes and prokaryotes for DNA replication. Okay. The resulting gaps are then filled by DNA pol 1 and DNA fragments are joined by DNA ligase, yielding an intact lagging strand. So one thing is that when you solve the questions, must note the questions is regarding either regarding the eukaryotes or it's regarding the prokaryotes, because the enzymes responsible for DNA replication for eukaryotes and prokaryotes is different. Okay. Next questions. Which statement is correct regarding the DNA replication in E. coli? Option 1. Both lagging strand and leading strand are synthesized towards the direction of replication fork movement. Option 2. Both lagging strand and leading strands are synthesized opposite to the direction of replication fork movement. Option 3. Lagging strands are synthesized opposite to the direction of replication fork movement, whereas the leading strands are synthesized towards the direction of replication fork movement. And the option 4 is leading strands synthesized opposite to the direction of replication fork movement, whereas lagging strands synthesized towards the direction of replication fork movement. The correct answer will be option 3, that is lagging strands synthesized opposite to the direction of replication fork movement, whereas the leading strands are synthesized towards the direction of the replication fork movement. Another important point regarding the lagging strand and leading strand synthesis is that leading strands synthesized continuously, whereas lagging strands are synthesized discontinuously. Here in the image it is seen that this is the direction of replication fork movement, and this is leading strand, this is lagging strands. The leading strand synthesized in the 5' to 3' direction in a continuous fashion, whereas the lagging strands synthesized towards in 5' to 3' direction in the discontinuous fashion. And it is the replication fork movement, and it is the direction of replication fork movement. Here it is saying that the leading strand synthesized is towards the direction of replication fork movement, whereas the lagging strands synthesized opposite to the direction of replication fork movement. Okay, and this image is taken from Brock Biology of Microorganisms. Okay, next questions. Which one of the following statements about DNA polymerase III of E. coli is not correct? Option 1 is it catalyzes NEET translation. Option 2, its absence is lethal to E. coli. Option 3, it synthesizes a complementary DNA strand using a single-stranded template. Option 4, it possesses 3' to 5' exonuclease activity. In option 2, it's mentioned that its absence is lethal to E. coli. It is correct because DNA polymerase III is the main replicative polymerase that helps in the replication. Whereas in option 3, it is mentioned that the DNA pol III synthesizes a complementary strand using a single-stranded template. It is also correct that because the DNA polymerase synthesizes a complementary strand and use a single-stranded template. Fourth option is it possesses 3' to 5' exonuclease activity. It is also correct because DNA polymerase III possesses 3' to exonuclease activity, which is the proofreading activity. Okay. So the answer will be option 1, that it catalyzes NEET translation, because DNA pol III does not possess the activity like the NEET translation. So this option 1, that is it catalyzes NEET translation, will be the answer. DNA polymerase III is the primary holoenzyme that involved in the replication activity. The DNA pol III holoenzyme also has proofreading capabilities that correct replication mistake by means of exonuclease activity reading in the 3' to 5' direction. The DNA polymerase III synthesizes DNA in 5' to 3' direction, because DNA synthesis always occur in the 5' to 3' direction. DNA pol III synthesizes base pair at a rate of around 1000 nucleotides per second. So the rate of nucleotides added is 1000 nucleotides per second. DNA pol III is the component of replisome, which is located at the replication fork. Next is about DNA pol II. DNA pol II possesses 5' to 3' DNA synthesis capability. It also has 3' to 5' exonuclease proofreading activity. DNA pol II interacts with multiple binding partners common with pol III in order to enhance its fidelity and processivity. Next, discuss about the enzymatic activities of DNA pol I. DNA pol I possesses a 5' to 3' that is forward DNA dependent DNA polymerase activity, requiring a 3' polymerase site and a template strand. 3' to 5' reverse, that is exonuclease activity, mediates proofreading. Next is DNA pol I also possesses a 5' to 3' exonuclease activity that mediates the NEET translation during DNA repair and primer removal. So this NEET translation activity is present in the DNA pol I. DNA pol I possesses the NEET translation activity. 5' to 3' RNA dependent DNA polymerase activity. DNA pol I operates on a RNA template with considerably lower efficiency than that does DNA templates, and this activity is probably of only limited biological significance. So from this we can consider that DNA pol III is the main replicative DNA polymerase, and DNA synthesis occur in the 5' to 3' direction. Both DNA pol 1, pol 2, pol 3 possess 3' to 5' exonuclease proofreading activity, and DNA pol 1 possess the NEET translation activity that helps in DNA repair and primer removal. Thank you. Please like, share and subscribe to this channel. Thank you.